Hi and welcome to this video. In this video, we will discuss gross brain very superficially, especially if you were in neuro. This is a much more superficial approach to the discussion of the brain. We'll spend the bulk of our time, though, talking about the ventricles, the scalp and dural reflections that create the dural sinuses, as well as discuss the eye. So there's a lot in this video. I do want to remind you, however, that if you took neuro, I want to caution you against pulling out all your old notes and going down the rabbit hole, because what we're going to cover is much more superficial since the details are covered in neuro. So here's a look at, at the brain, and notice that it has the bumps that are characteristic of the cortex. It has the bumps are the gyri. Singular is gyrus. The ridges or invaginations create these indentations or these ridges called sulci, and the singular of that is sulcus. In this view, what you see are the regions of the cortex, both the cerebral cortex shown colored, and the cerebellum is shown here, as well as the brainstem that is contiguous with the spinal cord here. The frontal cortex is in pink colored here. The parietal cortex is shown in blue, and separating these two cortices is the central sulcus. And it is the line shown here, and I'll show you another picture of that in a second. The temporal cortex is shown in yellow, and separating the temporal cortex from the parietal and frontal cortices is the lateral fissure, and that line is shown here, kind of a dashed line in red. The occipital cortex is in green, and that is that region that falls, kind of the landmark for that is that preoccipital notch that is present at the most anterior portion of this occipital region. Here's a picture from your Rowan where again you see the frontal cortex, the parietal cortex separated by the central sulcus. Then you see the temporal cortex and the occipital cortex, the lateral fissure here between the temporal cortex, parietal, and frontal cortices. If we now take a view through the one half of the brain, so if we sagittally section the brain, you will see again the frontal cortex is shown here, parietal cortex is here, and occipital cortex here. In this view, you see the parietal occipital sulcus, as well as the corpus callosum shown here. Now, inferior to that, so we're looking through a space called the third ventricle to the one of two thalami. So this is the thalamus shown here. And again, this view is through the third ventricle, which I'll describe a little bit more to you in a minute. Inferior to the thalamus is the hypothalamus, and then the pituitary gland sits here in the middle. What you can see on this image are portions of the brainstem, the midbrain, the pons, and the medulla oblongata. For some reason, I can't say that without thinking about the water boy. But anyway, so the third ventricle is here, and the third ventricle drains CSF, cerebrospinal fluid, through the cerebral aqueduct shown in red, to go to the fourth ventricle, which is this pink, somewhat fin-shaped structure right here. Again, so that's the third ventricle to the through the cerebral aqueduct to the fourth ventricle. And I'll show you some more of that in a minute. So when we are looking at these fluid-filled spaces that make up the ventricles within the brain, these spaces are responsible for making cerebrospinal fluid, because the choroid plexus, which is shown in red, is located in these spaces. So what I'm showing you here is a side view of the lateral ventricle. And the lateral ventricle has an anterior horn, which is uncolored, which I haven't colored here. So anterior horn, a body, an inferior horn, and a posterior horn. The body and the inferior horn contain choroid plexus, and thus contributes to the formation of cerebrospinal fluid, because in total, about 400 to 500 milliliters are made a day of cerebrospinal fluid, which bathe the brain, serve as shock absorbers, as a shock absorber for the brain. These lateral ventricles then are connected to the third ventricle. And remember, I told you that in the previous picture, the thalamus is located on each side of this third ventricle. The lateral ventricles, there are two of them, are connected to the third ventricle via this interventricular foramen, of Monro. And it shouldn't have an E on it. Okay. So through the lateral ventricles drain into the third ventricle via this foramen of Monro. And then notice that there is choroid plexus present in the third ventricle, which also makes CSF. From there, the CSF drains through the cerebral aqueduct, shown here, to the fourth ventricle. The fourth ventricle also contains choroid plexus, and then to exit the fourth ventricle, there are the foramina of Luschka. The foramen of Luschka is two of them. So each of them are lateral apertures. And then the median aperture is the foramen of Magendie, which is shown here, and then the central canal is here. Here's another view of those spaces. Imagine that a cast has been made so that those spaces are now have now become solid. So what you're looking at is the shape of those ventricles within those spaces within the brain. So here again are the lateral ventricles, and so the first and second ventricles, actually, they drain through those interventricular foramina to the third ventricle, then through the cerebral aqueduct to the fourth ventricle, and then the lateral apertures are shown here, those foramina of Luschka. In this image, you can get a more comprehensive view of the flow of cerebrospinal fluid, tracking just like we've discussed through the lateral ventricles, through the foramen of Monro, through the third ventricle, cerebral aqueduct, and fourth ventricle. Here at number 7 are those lateral apertures, and then the median aperture is here, that foramen of Magendie. In order to return to the venous circulation, there are extensions of the subarachnoid space that protrude into the dural venous sinuses. And so you can see here these tree-shaped or branch-shaped structures that project into the dural sinuses from the subarachnoid space. And this is in the area primarily of the superior sagittal sinus where these projections are made. So CSF travels or returns to the venous blood supply via these arachnoid granulations that are labeled number 10 here, that project into the superior sagittal sinus. And then eventually as we talk about the sinuses, you'll see that those sinuses then drain inferiorly through the internal jugular vein and then continue in circulation. So when you've seen the dura, we've talked about the meningeal dura, and you've seen the meningeal dura in the lab at this point, accompanying the spinal cord or surrounding the spinal cord. That meningeal dura is shown here in this kind of brownish color, and what's different or unique about the dura with the inside the skull is that there are two layers. There, in addition to the meningeal layer, there's a periosteal layer, which is tightly opposed to the bone, and the meningeal layer is shown here. The meningeal layer has reflections that create the sinuses, these venous-filled spaces, the superior sagittal sinus shown here, and then it continues separating the cerebral hemispheres more cranially. In the inferior portion of that is the inferior sagittal sinus. The reflections of the dura create this falx cerebri, and a falx is just a sickle-shaped structure, and it moves on the superior aspect of the cerebral hemispheres here. The arachnoid membrane is shown, I've shown here briefly in red, and that subarachnoid space is immediately deep to that. And remember those arachnoid granulations that I mentioned, which are ways that the cerebrospinal fluid is transmitted or returned back to the venous supply, project near the superior sagittal sinus, and you'll actually probably see the indentations of those arachnoid granulations on the dura as you work to remove the brain in the lab. Notice that the arachnoid membrane does not go into the sulci. However, the pia, which is shown in purple, does go into the sulci and stays tightly opposed to the brain. The dural reflections, the meningeal dura reflects to also create the transverse sinus that are on each side. And I'll show you some other sinuses that are created by the reflections of this meningeal dura. There is separating the cerebrum and the cerebellum is a dural reflection called the tentorium cerebelli, tent over the cerebellum, and it is shown here in green. So in this image, I just want you to pay attention to these two layers of dura. Again, the dura The meningeal dura separates from the periosteal dura to create, for instance, the superior sagittal sinus. And then deep to that is the arachnoid membrane. And notice these arachnoid granulations projecting into the superior sagittal sinus, as I described previously. This falx cerebri, falx cerebri is here in the inferior sagittal sinus, smaller than the superior sagittal sinus is shown here. Notice also in this subarachnoid space the presence of cerebral arteries and veins and in this subarachnoid space. So in the arachnoid granulations, the CSF, which has been circulating in the subarachnoid space, then gets projected or into the superior sagittal sinus, which is up here, and that CSF can flow both through cells and between cells to exit the arachnoid granulation and enter into the superior sagittal sinus. Recall that the emissary veins connect the superior sagittal sinus with the external portion of the scalp. Those scalp infections being able to be transmitted through these valveless emissary veins to these dural sinuses within the cranial vault. There are also these diploic veins that drain into the sinuses and are also valveless. The superficial cerebral vein located in the subarachnoid space also drains into the dural sinuses. We've talked about an extradural hematoma, and that extradural hematoma was caused, or the main cause of that is the compromising of the middle meningeal artery as it passes the pterion. These depression fractures at the pterion often lead to this rupture of this vessel and the extravasation of blood into this extradural space. And so we've talked about that. It is because it's arterial, it's under high pressure, and so often there is a brief loss of consciousness where the patient may regain consciousness and then later lose consciousness again, and it is potentially life-threatening. The subdural hematoma is the result often of the cerebral vein rupturing as it is entering into this superior sagittal sinus. And so this potential space between the dura and the arachnoid membrane becomes filled with blood, creating this subdural hematoma. The timing here is variable, and because it's under venous pressure, it's not as high pressure as, say, the middle meningeal artery that ruptured in the first example. The subarachnoid hematoma is the result often of a cerebral artery rupturing and then filling up this subarachnoid space with blood. And so we think of that as an aneurysm, and it can also be potentially life-threatening. I did want to remind you of the middle meningeal. Remember, we saw that when we discussed the infratemporal fossa, coming off the maxillary artery, traveling through the foramen spinosum, and then moving between the calvaria and the dura to supply the dura. Here again is another picture of those dural reflections. The falx cerebri here, that sickle-shaped structure, the superior part of which is the superior sagittal sinus, creates the superior sagittal sinus rather, and the inferior part is the inferior sagittal sinus. The falx cerebelli is here, also dividing the cerebellum into hemispheres, and then the tentorum cerebelli is here, forming a tint, a dural reflection kind of tint over the superior surface of the cerebelli. And anteriorly, this yellow part is the tentoral incisure. And so that's that anterior edge of the tentorum cerebelli. And then here you see that those tentorum cerebelli come together to form this diaphragma sella, which is goes over the pituitary gland. We've talked about the superior and inferior sagittal sinus, but there are other dural sinuses created by these reflections that also contain venous blood. So continuous with the inferior sagittal sinus here, you see the straight sinus, which joins the superior sagittal sinus and these transverse sinuses at the confluence of sinuses, the confluences of the sinuses. The sigmoidal sinus is this S-shaped structure here, and notice it's continuous with the internal jugular veins. The superior petrosal sinus is colored red, and then here's the inferior petrosal sinus. The inferior petrosal sinus communicates with this basilar sinus on the clivus, and then the cavernous sinuses are shown here, which communicate not only with the superior petrosal sinuses, but you'll see as we discuss the venous drainage of the orbit that they also contribute to or are connected to the orbital venous drainage, as well as being closely associated with the pterygoid plexus that we saw in the infratemporal fossa. Between the cavernous sinuses are intercavernous sinuses, and that's the only one that I had not yet mentioned. Now, I inserted this picture because while your notes do not specifically outline the, or there's no picture of the foramina in the skull that you are responsible for, I do want to draw your attention to, there is a chart in your notes where it talks about what is entering and exiting the skull. You are responsible for these foramina, and I want to just bring that to your attention via this image. Also, a structure that you're responsible for in lab, but are not, but is not discussed in your notes, is the arterial supply to the brain. Okay, and so the internal carotid artery enters through the carotid canal, and then the the middle cerebral and anterior cerebral arteries are branches of that. The anterior cerebral arteries connect with each other, and as one might imagine, it is a way for collateral circulation in the event that a portion may be damaged. The posterior communicating artery also is branching off of this internal carotid artery. Posterior cerebral is shown here, connecting to form this arterial circle. Okay. The basilar artery is here with the superior cerebellar arteries and anterior inferior cerebellar arteries, and then the vertebral arteries that go through those transverse foramina of cervical vertebrae, or through most of them at least, are shown here with the posterior inferior cerebellar. You are responsible for these arteries. However, your notes do not discuss those, so I want to make sure I draw your attention to that as well. So that was it. That is what we have for brain. We've covered superficial brain, the dural sinuses, ventricles, the arterial supply to superficial brain from the internal carotid artery. Next, we'll talk about the eye, and our discussion of that, of course, will be much more in depth. If we take a look at the bony orbit, notice that the orbits themselves are quadrangular pyramids with bases that extend anterior laterally. The medial walls are parallel to one another, and the eyes, when in anatomical position, are located such that the lateral aspects of the eye might be prone to injury. We've already talked about the muscles of facial expression and those that act on the eyelid, the orbicularis oculi, for instance. But now we'll get to talk a little bit more about the eye itself. Before we go any further into that, notice that the eyelid here, the upper and lower lids come together at the lateral corner, or the lateral canthus is what that is called. And the medial corner is called the medial canthus. There's also a lacrimal caruncle, or a piece of tough fibrous tissue that is located at the medial canthus of the eye that has within it some sebaceous glands that may assist in lubricating or allowing the eye ball to slide around or move around within the eye socket. The lacrimal puncta are shown here in yellow. Puncta is plural, and they represent, and so their function is to receive drainage of the lacrimal, the substance of the lacrimal gland, that is tears, that are washed from lateral to medial over the anterior surface of the eye. The white of the eye is called the sclera, and that tough fibrous material is continuous with the cornea, which is clear, that exists over the anterior 1/6 of the eye. The iris and pupil are covered by this clear cornea, where the iris acts to control the aperture or opening, which is the pupil. So dilating and closing as necessary to control the amount of light that enters the eye. The orbit itself is complex in structure in that it's made up of multiple bones: the frontal bone, the sphenoid bone shown here, the zygomatic bone, the maxilla, the lacrimal bone, and the ethmoid bone. And then there are several openings that are within the bony orbit. The optic canal, where the optic nerve enters the posterior aspect of the eyeball. The superior orbital fissure is shown here. The anterior ethmoidal and posterior ethmoidal foramina that we we saw and we talked about these nerves that go through these foramina when we were discussing the terminal branch of the maxillary artery as it went through the foramen to supply the lateral nasal wall and the nasal septum. Also, there is the lacrimal groove of the lacrimal sac and the inferior orbital groove, which, remember that one of the branches of the maxillary artery was the infraorbital artery, which went through the infraorbital groove and will exit the infraorbital foramen on the anterior surface of the maxilla. There are the bones in the anterior face, namely the frontal, ethmoid, sphenoid, and maxillary bones are pneumatized. In other words, they contain these air pockets that increase in mucus-lined air pockets that increased in space with age. So for instance, the frontal sinus becomes apparent usually around 7 years of age. The orbit is surrounded by these mucosal-lined air-filled spaces. And notice the position of the orbit relative to the frontal sinus, the maxillary sinus, and ethmoid sinuses. What's important is that the separation, the bony separation between the orbit and maxillary is pretty, maxillary sinus here is pretty thin. So blowout fractures, in other words, blows to the orbit from, for instance, a baseball or some object may displace the eye inferiorly, fracturing the inferior part of the orbit and causing the eyeball to retract and perhaps begin to invade this maxillary sinus area. Likewise, severe sinusitis of the maxillary sinus may, or a maxillary tumor, for instance, may erode this thin plate of bone and potentially begin to invade the orbit. Here's another image of the orbit, and in this image, it is filled with its bulbar fat and the eyeball. And then here is our maxillary sinus, and the small, the thin layer of bone that separates those two regions are apparent here. The other thing I want to point out in this picture is that the maxillary sinus, the drainage for the maxillary sinus is rather superior. And what's important about that is when you get sinusitis in the maxillary sinus, it can be difficult for that sinus to drain because of the superior location of its opening through the hiatus semilunaris. Hiatus semilunaris, which drains deep to the middle meatus. So on the lateral nasal wall, there are these shell-like invaginations called concha, and the spaces deep to those are meatuses. And deep to the middle meatus is where the hiatus semilunaris opens into the lateral nasal wall. When we take a look at the eye, I also want to draw your attention to, on this image, the orbicularis oculi, which again had that orbital part and a palpebral part. Remember, palpebral refers to the eyelid, and you can see that it also extends to the superior and inferior eyelids. The substance of that eyelid is made by this fibrous tarsal plate. There's a superior tarsal plate and an inferior tarsal plate. Those also contained glands that assist in the lubrication of the eyes and lipid secreting glands that help to prevent the tears from evaporating. The levator palpebrae superioris attaches to the upper lid and it elevates the upper lid. And we've already discussed the superior tarsal muscle. That was that small sympathetic innervated muscle that was responsible when the sympathetic chain in the neck was impaired for a partial ptosis or drooping of the upper eyelid. In red here, you'll see the conjunctiva. There is a palpebral and a bulbar part of the conjunctiva, and it meets at this fornix, this superior fornix. There is a corresponding inferior fornix. Remember that conjunctivitis is an inflammation of the conjunctiva that I'm showing you here in red. The cornea is that transparent anterior 1/6 of the eyeball, and then posteriorly, the posterior 1/6 is made up of the fibrous sclera, the white part of the eye. This eyeball, the eyeball is held in place in part by this fascial sheath of the eyeball, but also the inferior check ligament. And there are also lateral check ligaments that help to anchor the eyeball in the socket. The orbital septum here helps to connect the inferior eyelid, for instance, with the bony orbit. You'll also see the superior rectus here and the inferior rectus shown here. Both work to either elevate the gaze, or the inferior rectus depresses the gaze, but both muscles, the superior rectus and inferior rectus, adduct the eyes. So in other words, it moves the gaze toward the midline. The optic nerve is shown here. Notice that it is covered in dura, it's surrounded by dura. And the optic nerve pierces the posterior aspect of the eyeball. And then the bulbar fat, listed here as intraconal fat, same thing. A shock absorber, serving as a shock absorber for the eyeball, but also allows for accommodation of the different movements of the eye. Here's a color picture of that same image from the Grant's Atlas, and a close-up view of that superior tarsal muscle coming off of the levator palpebrae superioris. You can really appreciate these tarsal glands within the superior tarsal plate shown here, and then the orbicularis oculi, the palpebral part, also on the anterior surface of the eyelid. If we take a cross section of the optic nerve, as they've done here, notice that the optic nerve is surrounded by dura and arachnoid membrane and pia membrane. The running up along the center of the eye of the optic nerve is the central vein and artery of the retina. We'll talk some more about that as we continue to discuss the eye. So anchoring our tarsal plates in place, the superior and inferior tarsal plates, will be the medial palpebral ligament, the medial palpebral ligament and the lateral palpebral ligament. The orbital septum connected the inferior tarsal plate with the orbit. And then remember that the corner of the eye where the upper and lower lids meet the corner, the lateral corner was the lateral canthus, and the medial corner was the medial canthus. When we talk about crying, the lacrimal gland is nestled into a lacrimal fossa on the superior lateral aspect of the bony orbit. And so you will find that that gland with its 8 to 12 excretory ducts. You won't see the ducts, but you will see the gland present in gross dissection. You may also see accessory lacrimal glands. That is not uncommon. So when tears are made, they'll be secreted by these via these lacrimal ducts, and then the tears will move from lateral to medial, washing over the eye. The tears then leave the eye through the lacrimal puncta. Singular here is punctum, and through the superior and inferior canaliculi in order to then move to the lacrimal sac and the nasolacrimal duct, where excess tears will enter into the nasal cavity deep to the inferior concha. This is on the lateral side of the nose, and there were those shell-like projections that I mentioned previously when we were discussing the drainage of the maxillary sinus and the ethmoid air cells through the hiatus semilunaris, which is deep to the middle meatus. Tears via the nasolacrimal duct empty into this inferior meatus that's deep to this inferior shell-like structure on the lateral side of the nose. And then we would swallow our excess tears. The tear formation is a parasympathetic event where the parasympathetics arrive at the lacrimal gland, but they don't originate with the trigeminal branches that distribute those parasympathetics. We'll talk more about that, but it is via cranial nerve 7 that innervates, so the facial nerve, the parasympathetic GVE fibers that innervate crying originate in the facial nerve and then hitch a ride on or get distributed by branches of the trigeminal nerve in order to reach the target, which is the lacrimal gland. Sympathetics that accompany the internal carotid plexus are thought to inhibit crying as opposed to facilitate crying. So again, crying is parasympathetic, and the inhibition of crying would be a sympathetic event. So the muscles that act upon the eyeball are shown here. And so in orange, we'll start superiorly with the superior oblique muscle. This muscle courses through a pulley, also known as a trochlea, before attaching to the posterior lateral side of the eyeball. The superior rectus muscle is shown here, attaching to the superior part of the eye. Deep in this picture, on the medial surface near the medial part of the orbit, of the bony orbit, is the medial rectus. The lateral rectus has been cut, is here in purple. And then the inferior oblique is shown here in red, and the inferior, I'd said the inferior rectus already, I think. Anyway, the inferior rectus. And then this is just a superior view of these muscles. So in this image, you can see the nerves that innervate these ocular muscles. In purple, you see the trochlear nerve, which is cranial nerve 4. The trochlear nerve innervates the superior oblique muscle, which goes through a trochlea, changing the direction of its pull before it inserts on the superior aspect of the eye. The nerve shown here in green is the abducens nerve, and the abducens nerve innervates the lateral rectus. The lateral rectus AB ducts the eye, abducens, abducts the eye. All of the rest of the ocular muscles, the superior oblique, I'm not the superior oblique, the superior rectus, the levator palpebrae superioris, the medial rectus, the inferior rectus, inferior oblique are all innervated by branches of the oculomotor nerve that are shown here in orange. The superior branch, of course, goes to innervate those superior muscles, superior rectus, levator palpebrae superioris, and the inferior branch or inferior, you may also see inferior division, goes to the medial rectus, inferior rectus, and inferior obliques. Also shown on this image is the trigeminal nerve. So here's the trigeminal ganglion where my pointer is, and it's 3 divisions. It's ophthalmic division, its maxillary division, and its mandibular division coming off of it. Hanging off of the maxillary division in the pterygopalatine fossa, which we saw in the infratemporal fossa, is a parasympathetic, one of 4 parasympathetic ganglia called the pterygopalatine ganglia. So parasympathetics from the facial nerve go to the pterygopalatine ganglia. They synapse and then postganglionic parasympathetic fibers then hitch a ride on branches as trigeminal to get all the way up to the lacrimal gland to innervate crying. Remember that the first and second divisions of the trigeminal nerve, the ophthalmic and the maxillary nerve, its branches are purely sensory, but the parasympathetics, so GVE fibers from the facial nerve that originated with the facial nerve are carried to the target, the lacrimal gland, by those sensory branches of the trigeminal nerve. This image shows you a little bit more about those divisions. You can see the maxillary nerve going through the foramen rotundum, and then its branches moving anteriorly through the face, right? And then the ophthalmic branches coming off of the first division of the trigeminal then are going into the orbit where sensory branches will become important. If we take a closer look at the muscles and how they act upon the eye, shown here is the superior rectus. And I told you that the superior rectus and inferior rectus work to adduct the eye, so to move the gaze medially toward the nose, for instance. Notice that they attach, the superior rectus attaches superiorly, and the direction of its pull, it will both adduct and elevate the gaze. The inferior rectus will depress the gaze, but also adduct, and then the medial rectus will pull the eyeball medially, also adducting the gaze. In contrast, the superior oblique, the inferior oblique, and the lateral rectus oppose those actions, adducting the eye. In addition, the superior oblique also depresses the gaze, whereas the inferior oblique elevates the gaze. Notice the direction of pull of these muscles, and that'll become important as we talk about the clinical testing of those eye muscles, as you start to think about clinical testing of those eye muscles. For rotation of the eye, we're talking about those that cause the eye itself to move in either a clockwise or counterclockwise manner. So intorsion would be medial rotation of the eye, and extorsion would be lateral rotation of the eye. So in summary, when we talk about the movements of those muscles on the eye, the superior rectus, for instance, elevates and medially moves the eye medially, adducting the eye, whereas the inferior oblique elevates the gaze and also laterally moves the gaze laterally, or abducting the gaze. The lateral rectus and medial rectus are apparent, and then the superior oblique depresses and laterally moves the gaze laterally, abducting the gaze, and then the inferior rectus adducting the gaze. To clinically test the muscles, it's necessary to move the eye to separate, for instance, the actions of the superior rectus and the superior oblique. This is a summary of what needs to happen in order to test, for instance, the superior oblique. The superior rectus. We would need to instruct the patient to avert the gaze laterally and then up in order to test the superior rectus. Likewise, for the superior oblique, we would have to instruct our patient to avert their gaze medially and then to depress the gaze in order to test the superior oblique. I will post a short optional video which will explain to you how that clinical testing occurs, and so I suggest that you familiarize yourself with that. But here the information is in summary. As we take a closer look at the eye, we've already talked about the sclera, this outermost layer of the eye. We think of those as tunics or coats, and the sclera and the cornea make up the outer tunic of the eye, the external fibrous tunic of the eye. The middle tunic includes the choroid layer, and the choroid layer, shown here in purple, contains arteries and veins that supply the eye, as well as the ciliary body, shown here in purple, and the ciliary processes that are also present. The zonular fibers extend from the ciliary processes, also called suspensory ligaments, and they act upon the lens, either increasing the tension or allowing it to round, or decreasing the tension, allowing it to flatten out to change the focusing of the gaze. The middle tunic, the internal tunic, or the innermost coat, is the nervous layer. It contains the retina, and I've drawn it in yellow here. It is on the posterior part of the eye, and it is responsible for detecting these changes in light and color via the rods and cones that are located within it. The chamber here is the vitreous chamber. It's filled with this gel-like substance, the vitreous humor, and that is in contrast to the posterior chamber, which is shown in blue, and the anterior chamber, which contain aqueous humor. And the aqueous humor is made by these ciliary processes, secreted into this posterior chamber, and then it moves into the anterior chamber, and then exits the eye through the sinus venous sclera, and we'll talk a little bit more about that in a minute. When reading, so reading is a parasympathetic event, and so this parasympathetic event causes the contraction of the ciliary muscles, which then in turn, kind of counterintuitively, allows this lens to round up so that we can converge the gauge, focus for accommodation when reading. So reading is an example of a parasympathetic event. Also, in line with that is the constriction of the pupil as a part of reading. And so the sphincter pupillae, shown here in red, would contract to assist in this accommodation process. In contrast, the dilator pupillae muscle, which would be these spokes, these yellow spokes on the iris, would dilate when they contract in a sympathetic, via sympathetic innervation, would dilate the pupil, enlarging the aperture so that more light, for instance, would enter into the eye. I mentioned to you about the aqueous humor being made by these ciliary, secreted by these ciliary processes, and the aqueous humor goes into the posterior chamber, then to the anterior chamber where it drains into the sinus venous sclera, which is this canal of Schlemm. So here is the full path where the ciliary processes make the aqueous. It goes to the posterior chamber, then the anterior chamber, through the spaces of Fontana to the canal of Schlemm, or the sinus venous sclera. It's from there that they reach the aqueous vein and anterior ciliary vein to return to the venous system. A blockage of anything along this process where the aqueous humor increases because it's not draining effectively, for instance, leads to glaucoma. And so an increase in this pressure of the eye that is potentially damaging and could lead to glaucoma. Here's just another image of that where the cilia, if you follow this dashed line, the ciliary processes make the aqueous from the posterior chamber to the anterior chamber. And again, the sclerovenous sinuses to return via the ciliary veins back to venous supply. In this slide, you can see the arterial supply of the eyeball, and the arteries and veins that are embedded in that choroidal layer originate with branches off of the internal carotid artery. So briefly, the internal carotid artery gives off the ophthalmic artery, which rides deep to the optic nerve for a period of time before giving off the central artery the central artery here of the retina, which pierces the dura, enters into the optic nerve, and then travels towards that to terminate at the inner tunic. The ophthalmic artery also gives off short posterior ciliary arteries, which pierce the posterior part of the eyeball and supply only the posterior part. And there are multiple short posterior ciliary arteries. The ophthalmic artery will give off a lacrimal artery, which anastomoses with the middle meningeal artery that came off of the maxillary artery, a branch of the external carotid artery. And then the lacrimal artery also communicates with the anterior ciliary artery, which goes to the anterior surface of the eye, pierces the sclera to then access the choroidal layer and anastomose with the long ciliary arteries that came also off of the ophthalmic artery. The ophthalmic artery also sends off a supraorbital, which goes through the supraorbital foramen on the anterior superior aspect of the face, and then the anterior and posterior ethmoidals, which we saw anastomosing with the sphenopalatine branch of the maxillary artery in the lateral nasal wall and the nasal septum. The nasal artery anastomoses with branches of the facial artery on the anterior part of the face, and the supratrochlear artery, which also goes through the supratrochlear foramen. Here's another image of those short posterior ciliary arteries, and you can see them piercing the sclera to go to, this picture is a little bit misleading in that these are located in the choroidal layer, that middle tunic. And so the short ciliary arteries are here going to the posterior aspect. The long ciliary artery anastomoses with the anterior ciliary artery through either this major arterial circle or the minor arterial circle, providing extensive blood supply to the anterior eye. Also shown on this image are vortex veins, which drain the middle tunic, and these vortex veins then connect with the inferior ophthalmic vein or the superior ophthalmic vein to then drain the eyeball. We've already talked about that. I've already shown you that. In this picture, though, I want you to realize that the central artery of the retina is an end artery. In other words, it terminates shortly after reaching the inner part of this retinal layer, that inner tunic. After going through this cribriform plate. And then the central vein of the retina would drain that parts of the posterior parts of the eye. What I like about this image is that you can really appreciate the extensive arterial and venous layers of vessels in this middle tunic of the eye, this choroidal layer, this choroid layer of the eye. Here again are this vortex or verticose veins. You'll see both of those words in different texts. And then you also can see the the arteries and venules that are going to help to supply this retinal layer, and I'll talk a little bit more about that. What I have not drawn your attention to is the fact that here's the optic disc. And so this is where the optic nerve enters the eye. It is a blind spot in the region of least acuity, as there are no rods and cones located in this particular area. The part of the retina that is of highest acuity is the macula, and within it is the fovea centralis. The fovea centralis has the highest concentration of cones, and thus is the site of highest acuity. You can see the macula here, and then this light spot is the optic disc in this image of the eye. The short posterior ciliary vessels are shown here, piercing the sclera of the eye to gain access to the choroid layers, and the central arteries and veins of the retina are riding inside the optic nerve towards that inner tunic where it can help to supply the retina. Those vortex veins that we observed drain, as I said, into the inferior and superior ophthalmic veins within the bony orbit. Notice here the extensive communication with the cavernous sinuses, the pterygoid venous plexus in the infratemporal fossa, the angular vein and facial vein on the anterior aspect of the face, the supraorbital vein, which will go through the supraorbital foramen. So there's extensive connections and significant path for ocular infections that get unmanageable, for instance. I don't know why I put that picture in there. When we talk about the posterior eye, and we talk about the location of structures in the eye, we refer to it as superior, inferior, temporal, or nasal. And so you would describe it in those terms. For instance, the optic disc is nasal to the macula, which is located over here, the site of highest acuity, the site of lowest acuity shown here. As such, the photoreceptor layer and the nervous layers can become detached from each other, which is a retinal detachment, resulting in floaters that are blood droplets that may float in and out of the field of vision. The ocular muscles, the superior rectus, medial rectus, inferior rectus, and lateral rectus, originate at this common tendinous ring. And so this yellow line that I'm showing you here is a common attachment for these muscles. The optic nerve is shown here with the ophthalmic artery. Remember, it rode along deep to the optic nerve for a portion of time before then looping over it to give off other branches to supply the superior orbital, for instance, or the anterior and posterior ethmoids, for instance. Within the common tendinous ring, there are also structures that pass through the common tendinous ring or inferior or superior to that via the superior orbital fissure. And so the superior orbital fissure was on the posterior aspect of the bony orbit, and those structures that are superior to it, it's important to know which structures are superior to it, within it, within the common tendinous ring and inferior to it. So the trochlear nerve, the frontal nerve, and lacrimal nerve, lacrimal and frontal being a part of the first division of the trigeminal nerve, and the trochlear nerve that'll go on to innervate the superior oblique. Those exit or enter the orbit superior in the superior part of the superior orbital fissure above the common tendinous ring, along with the superior ophthalmic vein that we saw in previous slides. Within the common tendinous ring, you'll see the superior and inferior divisions of the oculomotor nerve, as well as the nasociliary nerve, which is part of the trigeminal nerve, branch of the trigeminal nerve. The abducens nerve also is within the common tendinous ring, and you see it here coming out at you, going to supply the lateral rectus. And finally, the inferior ophthalmic vein is the only thing that is inferior to the common tendinous ring, and it's still within the superior orbital fissure. Here's another view of that, I think, that helps you to visualize this common attachment for these ocular muscles and those structures that are exiting the superior orbital fissure, both within and outside of that. Here's an anterior view, and sometimes we ask the TAs to do an anterior view of the orbit, and so this is what that will look like once the eyeball has been removed. So first I want to talk about the trochlear nerve. This is cranial nerve 4. It has general somatic efferent fibers within it. So here's our trochlear nerve going to our superior oblique muscle. And remember, the oculomotor goes to all of the other ocular muscles, with the exception of the lateral rectus, which is supplied by the abducens, which is cranial nerve 6. Here's the location of the trochlear nerve in the superior orbital fissure above the common tendinous ring. And then note the location of the oculomotor nerve and the abducens. Before entering into the superior orbital fissure, the oculomotor nerve, the trochlear nerve, and the abducens nerve, they all travel through the cavernous sinuses, which were these venous-lined spaces that were located, that we talked about when we talked about the dural sinuses. The internal carotid artery travels through the cavernous sinuses, and it's thought that the cooler temperature of the venous blood may help to regulate the temperature of the arterial blood as it is entering into the cranial vault. So here's the trochlear nerve located in the cavernous sinuses. I want to draw clinically your attention to aneurysms of the internal carotid artery may impact this trochlear nerve, and because it impacts the trochlear nerve, it may affect the functioning of the superior oblique muscle. And so you will notice that as the superior oblique laterally or abducts the eye and also depresses the gaze, those functions may be impaired or weak if either by aneurysm or a infection in the cavernous sinuses may, for instance, compress the trochlear nerve. The same is true of the abducens nerve and the oculomotor nerve, and we'll talk a little bit more about that. Here again is a reminder of where those cavernous sinuses are located, shown in brown, just for you to refer back to so that you can understand what I'm talking about when we say the cavernous sinuses. There are extensive connections, and when we talked about venous drainage of the orbit, of the bony orbit, remember that the cavernous sinuses are connected to that. And so an infection in the cavernous sinuses may not just infect, for instance, the structures within those sinuses, but also it may affect the eye or result from collateral circulation around the orbit. Here's the abducens nerve. Remember, it innervates the lateral rectus in order to check the lateral rectus. So if the lateral rectus, for instance, is impaired, then the medial rectus may be unopposed, and so a medial deviation of the gaze may result. So that's an example of what would happen if, for instance, the abducens nerve is impaired in the cavernous sinus. Here is oculomotor. Not only does the oculomotor nerve, cranial nerve 3, provide innervation via general-somatic efferent fibers to the ocular muscles, with the exception of the superior oblique and lateral rectus. But the oculomotor nerve also carries or has parasympathetic GVE, general visceral efferent fibers that are associated with it. Those GVE fibers are going to the optic ganglia, which we will see hanging off of that first division of the trigeminal nerve. So those ophthalmic branches of the ophthalmic nerve, you'll see the ciliary ganglia there. And from there, postganglionic parasympathetic fibers will go to the ciliary processes and the sphincter pupillae, resulting in rounding of the lens as the ciliary muscles contract, and also constriction of the pupil via the sphincter pupillae muscle. Those cranial nerves, the oculomotor, trochlear, and abducens nerve are shown here. You are responsible for those cranial nerves in lab at the base after the brain has been removed from the area. And so I'm just taking a moment to point out those three to you, but you can learn the rest of those on your own. Again, those parasympathetics associated with the ciliary ganglia originate from the oculomotor nerve. So those preganglionic parasympathetic fibers travel to the ciliary ganglia, which then there they synapse, and then postganglionic parasympathetics then travel through the short ciliary nerves to get to the dilator, the sphincter pupillae muscle and the ciliary processes. In contrast, the sympathetics, which originated in the IMLCC, T1 to L2, they, the more cranial parts, in T1, they preganglionic sympathetics travel to the superior cervical ganglion, where they synapse, and then postganglionic sympathetic fibers surround the branches of the internal carotid and then often also get distributed by branches off of the trigeminal nerve. And so you see here that the long ciliary nerves, which is part of the first division of the trigeminal, distribute the sympathetics to the dilator pupillae muscle. So we've talked extensively about the maxillary and mandibular divisions, or the second and third divisions of the trigeminal nerve, when we discussed the infratemporal fossa, but what we haven't discussed are the branches of the first division of the trigeminal, which is the ophthalmic division. After or as the ophthalmic nerve goes through the superior orbital fissure, it very quickly or just proximally gives off the lacrimal nerve. The lacrimal nerve is a sensory nerve, but remember that the parasympathetics from the facial nerve get distributed to the lacrimal gland via the lacrimal nerve. So again, those parasympathetics aren't a part of the trigeminal nerve. Those originated with the facial nerve and are simply opportunistic in trying to get to the lacrimal gland. Also off the ophthalmic is the frontal nerve, which gives us supraorbital and supratrochlear nerves, where you can expect that the supratrochlear nerve will be close to that pulley or trochlea that redirects the pull of the superior oblique muscle. The nasociliary is shown here with the ciliary ganglion, one of those four parasympathetic ganglia that are associated with the head. We talked about the pterygopalatine ganglia that in the pterygomaxillary fissure, in that space in the infratemporal fossa, and the otic ganglia was associated with the parotid gland, innervation of the parotid gland via the glossopharyngeal nerve. The ciliary ganglia is shown here, and the short ciliary nerves that are part of the trigeminal nerve, they distribute parasympathetics from the oculomotor nerve that we just talked about. And so that is here. Notice also the long ciliary nerve. Recall from the previous slide that the long ciliary nerve distributes the sympathetics that originated in the IMLCC, and those preganglionic sympathetic fibers ascended in the sympathetic chain to the superior cervical ganglia, and then postganglionic sympathetics accompanied the internal carotid artery till it got to near the orbit, where the long ciliary nerve, where those branches then get to the eye via the long ciliary nerve. The anterior and posterior ethmoidal nerves are shown here, sensory to the lateral nasal wall and nasal septum, for instance, and terminated in the external nasal branch and the infratrochlear branch. The external nasal branch going to the anterior face near the nares, and I will talk about that a little bit more later. Yep, we said that. All right. Again, what I like about this picture is that you can really appreciate the wheel and spokes, where the dilator pupillae are the spokes that help to pull open the aperture, making the pupil larger. The dilator pupillae is sympathetic. Innervation has sympathetic innervation. In contrast, the constrictor pupillae, these circularly arranged muscles, they contract the pupil, making the diameter smaller. These are parasympathetic in nature. The oculomotor has both GSE and GVE nerve fibers in it. You do not need to know the oculomotor nucleus. You do not need to know the nuclei of these cranial nerves. However, Edinger-Westphal nucleus is one of the few nuclei that I do need you to know. The oculomotor nucleus, so those GSE fibers that innervate those ocular muscles, with the exception of the lateral rectus and the superior oblique, those are GSE fibers that are innervating those muscles. The parasympathetics originate in this Edinger-Westphal nucleus, and then those preganglionic parasympathetic fibers travel to the ciliary ganglia, where they synapse, and then postganglionic sympathetics via the short ciliary nerves then go to the sphincter pupillae and the ciliary processes, just as we've discussed before. And then finally, I want to remind you of Horner's syndrome, right? That was ptosis, anhidrosis, meiosis. And we talked about it when we discussed the neck, the sympathetics in the neck. And this image really just highlights that Horner syndrome again for you.